Welcome everyone to this If Oxford event, Chemistry Confections, the sweet version. It's a pleasure to introduce Jess Crompton and Jonas Lodge. Jess, you remember from last time, is an experimental chemist at the University of Oxford, and Jonas Lodge runs and owns a restaurant called GL50 in Cheltenham. So we have Jonas's kitchen and Jess's periodic table as well as part of the show. So Jess, over to you.
Okay, thank you, Dane. Okay, I'm going to start us off, so I'll share my screen. Brilliant. Okay, so as Dane mentioned, I'm Jess Crompton. I'm an experimental organic chemist or a synthetic organic chemist at Oxford University. I'll come back to what organic chemistry is in a little while. I'm in the second year of my PhD, and I also studied my undergraduate degree at Oxford. So moving on to sort of how do I view chemistry in everyday life? So yes, chemistry is in the lab and all of our sort of traditional science images, but there's also really important aspects of chemistry and impacts of chemistry as the science in lots of other areas of our lives. So we've also massive impact of chemistry in the oil industry, crude oil industry, in the fast-moving consumer goods industry. So, for example, cleaning products, in the pharmaceutical industry, even in makeup and skincare, paints for pigments, and the power industry in the form of batteries. But what we're going to look into today is something that we use every single day without fail, otherwise we wouldn't be very healthy, is the chemistry and the science behind the food that we eat. This is more of a savoury image, but today we're doing the sweet version, as Dana said. So we're focusing on a lemon meringue cake. So a little bit of a crash course in organic chemistry for people. We'll start with the sort of basic, most iconic image of chemistry that people might have, and that would be the periodic table. So here we have it. Once upon a time I had to learn this, but it's gone from my brain now. So as an organic chemist, I am interested in the chemistry of carbon. So everything else, it looks a bit strange. I'm just interested in one element of all of them, but believe me, there's a lot of diversity we can get from carbon. So it's carbon and the atoms attached to it. And most of the chemistry that we're going to look at today is going to be organic chemistry, so will be sort of carbon centric, but we will encounter some other elements as well, mainly hydrogen, which is carbon's best friend, nitrogen, oxygen, and we might have a little bit of phosphorus and sulfur sneaking in there as well. The other elements are much less common in sort of biological systems, and by virtue of that in our food. We will see a little bit of sodium a little bit later, but mainly we're looking at organic chemistry today. So as organic chemists, we like to draw the carbon atoms and how they're attached to other atoms within the molecule. And the way that we do this is we explain that they are bonded together, and that's how we get chemical diversity. So here I have a model of exactly what I've got drawn on the screen. So you can see here I've got a carbon joined to a carbon. So these are the two black atoms here joined together. I've got 3 hydrogens on the left-hand side carbon. These are the little white atoms. I've got 2 oxygens. One of them has a double bond. That's the 2 bendy bonds I've got on my model. One has a single bond. That's the straight one. And an oxygen on the end. So this is what an organic chemist would draw sort of to explain what they mean. However, we're inherently lazy and we draw lots of very big molecules, much bigger than this is just vinegar or acetic acid. So we have a bit of a shorthand, and I'm going to use the shorthand because I'm an organic chemist and I'm a bit lazy. So the way that we have this shorthand is we say that if the carbon has nothing else attached to it, or anything that's sort of not clearly attached to it. So this carbon here is only drawn attached to another carbon. We don't draw the carbons because they take time, but it's only attached to one carbon. So what we say is that the other atoms on this carbon must be hydrogen. So we sort of imply the hydrogens, And we still draw the oxygens, the nitrogens, the phosphoruses, everything else we still draw, and we'll still draw hydrogens if they're attached to those heteroatoms. Heteroatoms are atoms other than carbon. So hopefully that makes sense to everybody. We also have a shorthand for 3D molecules. So molecules aren't flat, they are three-dimensional. Unfortunately, I ran out of atoms, so I've had to make do with this other small set. So this is menthol. And if I think, is that the right way around? So you can see here that this is not a flat molecule. There is a lot of diversity. There's also this sort of cartoon here, if you're struggling to see this model. But you can see that I've got some bonds that are these sort of wedge bonds and some bonds that are these hash bonds. So the wedge bonds mean that the group of atoms or the atom is coming out of the page or out of the screen towards you. So that would be this group of atoms that I'm spinning around here. And the hash bonds mean that it's going sort of into the plane of the page or the screen. So that would be, it was coming towards me, as you can see, this group here, because coming out is this hydrogen here. Hopefully that makes sense. I'm sorry that this is not such a nice model as this is, but I have some nicer models for you later. Okay, so we also get diversity within organic molecules as a result of functional groups. And functional groups are sort of the way atoms are bonded together to give us certain properties of a group, if that makes sense. So we've got different functional groups. We've got alkanes and alkenes. These are really common in really oily molecules, similarly with arene groups. We have alcohol. Now that's not the alcohol that is in wine necessarily, although ethanol is in wine and is an alcohol, but any molecule that has an OH group would be classified as an alcohol, like this. By the way, R is just any sort of generic group. It's a way of chemists sort of saying this is not the interesting bit that we want to focus on. So R is just another carbon containing group. Amines are kind of the nitrogen analog of alcohols, but because of it being a nitrogen, not an oxygen, they have specific properties. And there's various others. These are all carbonyl compounds, so they all have this sort of common carbon double bonded to oxygen motif. And each one of these functional groups has sort of specific properties. We'll come back to some of these later, but all being different means that they react in different ways. And they also are responsible for the sort of physical properties of the molecules as well. So in terms of physical properties, it might be in terms of the bulk property of the material. So whether something's going to be a liquid or a solid. Equally, how it would react with other bulk material, for example, with water. So some functional groups are inherently hydrophilic. That means they love water. And will happily dissolve in water if they contain lots of these functional groups. These are some examples of these types of things. So anything that has a charge or these types of functional groups will generally be hydrophilic. Hydrophobic are those things that I mentioned as being greasy. They are sort of more oily, don't like to be around water and are hydrophobic. We will come back to these terms quite a lot today. Also, functional groups are responsible for the sort of chemical characteristics and which reactions can take place. So here's a very important reaction for our bodies. We take carboxylic acids and an amine, and that forms an amide bond or a peptide bond, which is one of the key bonds in proteins in how they are made up. So if we're going to talk about the chemistry of food, we need to know a little bit about the chemistry of basic food compounds. So hopefully everyone's familiar with the three sort of main food groups, being carbohydrates, proteins, and fats. I'm just going to have a quick sort of talk through the chemistry involved here. So carbohydrates are polymers, and that means that they're made up of lots of small subunits, or what we would call monomers. And they're joined, they can be joined in a long chain, as you can see with the dark blue hexagons. They can be branched, as you can see with these light blue hexagons popping off my chain. And the monomer is generally a simple sugar. This is a simple sugar. I've also got a model of one. Please pretend that this yellow atom is red. But this is a simple sugar. This is glucose, and that's the molecule that you see on your screen. You can see that these are not flat and that there is a sort of representation that we use If I do it that way, that's the same way that you can see it, I think. So these are the monomers that are all joined together to make carbohydrates. Proteins, similarly, are polymers. However, they have a different monomer, so a different subunit. And their subunits are amino acids. So thinking back to those functional groups I've talked about, we've got a carboxylic acid group at one end, we've got an amine group at the other end, and then we have this R group. We'll keep talking about this R group today. It's basically, there's about 20 naturally occurring R groups. They all have different properties, and that can have some important effects, especially in baking. But as these are all joined together, they're joined into a long chain or a polypeptide, and then this chain can sort of fall back on itself, and we get interactions between different parts of the chain, and that gives us this sort of specific 3D structure that you can see on the left-hand side of the screen. And now with proteins, the structure of proteins is really, really important and integral to their function. So if we lose the structure and the 3D structure of the protein, generally it doesn't work anymore. And that is called denaturation, and we'll come back to that as well. So finally, we're getting to the end of this sort of crash course section. So fats are not polymers. They are actually molecules. They're big molecules, but molecules nonetheless. And they basically look like this. So you have one sort of linking group, and then 3 long chains coming off. These are called triglycerides, these molecules, and they're made up of glycerol, which is your linking molecule, and 3 fatty acids. There's different types of fatty acids. There's saturated fatty acids, unsaturated fatty acids. This one's monounsaturated, and this one's polyunsaturated. These fatty acids are linked to whether you have saturated fat or unsaturated fat, and determined by which types of fatty acids you have. So if I put that together in a molecule, that's kind of what those molecules will look like. Also, an important type of fats within our bodies are phospholipids. So these have an additional component. So we lose one fatty acid chain and we add a phosphate head. Now, these are quite interesting because this phosphate head is hydrophilic. The fatty acid chains, as I said, alkane and alkene functional groups are really greasy. So the fatty acid chains are hydrophobic. The phosphate head is hydrophilic. So this molecule has like 2 characteristics. It's what we call amphiphilic, because it displays both properties. And so it sort of has strange bulk properties. So you can see here, this is a cartoon version, but in a watery environment, they'll form these bilayers. So the dots or the purple circles are the phosphate heads, which are hydrophilic and will happily interact with the water molecules, whereas the fatty acid tails are sort of sheltering in between the 2 phosphate heads. And then if you've got a mixture of oil and water, you'll form these sort of micelles or globular structures here, where you can see if you had water on the outside, the phosphate heads will stick into the water and the fatty acid tails will remain in the oil, maximizing the favorable interactions. Okay, so that's our sort of crash course into food and food compounds done. So today we're going to look at the chemistry or the science of baking through a key bake. And today we're going to look at a lemon meringue cake. So we're going to start off with a structure and a cake structure. Hopefully everybody's made a cake before, even if it's one out of a packet. And one thing that sort of is quite interesting and hopefully works in your cake, otherwise it's going to be not a very nice cake, is when your batter goes in the oven, it rises and gives us a fluffy texture. But why? And the reason is because we add a raising agent. Now, this can be in the form of many different things. We can add self-raising flour. We can add bicarbonate of soda. And that's also baking soda. It's the same thing. Or baking powder. Now, the thing that all of these 3 raising agents have in common is they all contain sodium hydrogen carbonate, or bicarb, which is bicarbonate of soda. It has lots of names because it's sort of quite old and, well, no, it's a traditional chemical, so it has lots of different names that have been sort of passed down. And what happens is when we heat this up, so in the oven, the sodium hydrogen carbonate, or the bicarb, will decompose, and it gives us some sodium carbonate, which is harmless and just sits in your cake. It gives us carbon dioxide and water. Now, carbon dioxide is a gas, and water, because we'll be above 100 degrees in the oven, that will also be a gas. So this gives us gas bubbles. And then because of the heat of the oven, these inflate. And that means that our cake batter rises. And also the batter's kind of setting around it, which means the bubbles don't just sort of bubble out and escape, but they make the batter rise. So this reaction is called a thermal decomposition reaction. But one thing that sort of I was always like, what's the difference between bicarbonate of soda and baking powder? Because sometimes you look in the cupboard and you've only got one, and you think, well, can I substitute it? And actually, they are slightly different in that baking powder also contains something called cream of tartar, which you also can buy separately, just to confuse matters. Now, this is cream of tartar. This is its structure. It's an acidic salt, and that's because it has this carboxylic acid group here. So it can release a proton, or H+, and that's what makes things acidic. And since bicarb is a base and cream of tartar is an acidic salt, when you add it to water, the acid dissolves and then you get a reaction happening. So the reaction that happens, you add acid to your bicarb, you get sodium salt. We don't really worry about that. But again, we're getting the carbon dioxide and water. And again, that generation of gas bubbles. Now, this reaction does go at room temperature, so you will get actual water, but in the oven, when it happens, you'll get water as a gas. So again, we're getting bubbles which inflate the mixture. And generally using baking powder rather than baking soda or bicarb alone, because you get the carbon dioxide released over a wider temperature range, because this reaction is a bit more reliable than a thermal decomposition, it means that you get a more reliable rise in your cake, and probably you can bake it at sort of a more varied range of temperatures and it will still work for you. So just to sort of illustrate what I've said there, I'll just lose my laser pointer so that I can click. Here we've got 2 glasses. I promise you, in each glass is just water. So I'm going to add baking powder and bicarbonate of soda to it. So you can see as soon as I add it, the baking powder starts bubbling. Now remember, that's because we've got the acidic salt, which dissolves in the water and then reacts with the basic bicarb. So it's to release carbon dioxide, and that's what these bubbles are. In contrast, we've got the bicarbonate of soda. There's no acid in the water, so there's no reaction taking place there. Hopefully everybody could see the bubbles. Just to sort of prove to you that the bicarbonate of soda does contain sort of contained gas that can be released, and it would be released in the thermal decomposition. I'm just going to add a little bit of external acid. This is just vinegar. Lovely, this has got sound. So you can see that as soon as I add the vinegar, all of that gas is released. So you can see that the bicarbonate of soda does sort of will release gas, but doesn't release gas until it's sort of prodded to. So here we've added acid, and in a cake you heat it up and that causes the thermal decomposition. And I clearly had a lot of fun making this video and kept adding more and more vinegar. Okay, so that's not the only ingredients we've got in cake. It would be quite boring if we just put raising agent in cake. So we've also got eggs. So eggs contain protein, which denatures. That's when the proteins lose their 3D structure, and I'll come back. And what it does is it sort of coagulates in the cooking process, and it gives us this final firm texture. So proteins generally are quite globular things. They're all quite discrete, as you can see here with my paper clips in a mug. When you're just at sort of physiological temperature, room temperature, they'll look like this. And then as you heat it up, there's sort of lots of weak bonds that hold the specific 3D structure together break, and basically the protein unravels a bit, and it basically means that they have a looser and more random structure, and they can tangle with each other. So as you can see here, I've heated up my proteins and my paper clips, and that means that the weak bonds holding them together have broken, and then they've sort of got a more loose and random structure, and that would be the coagulation. Also, we add sugar to our cakes. Now with cake, sugar is mainly just a flavouring agent. Not that much to say, more to say about sugar in this case. Butter is an anti-staling agent for cakes. And the reason that cakes would go stale is because you get the migration of moisture from starch granules within your cake. And this degelatinizes the starch. And then you get some of the starch molecules will recrystallize and all join up. And that gives you a hard texture. But if you separate the starch molecules using butter or a fat, that keeps them further apart. And it means that they can't recrystallize as much. So you get less staling. Also, you add flour to your cakes, as well as some flours such as self-raising flour containing a raising agent. All flour contains gluten. If you were here last time, we talked a lot about gluten and the gluten development in kneading dough for our pizza. But for anybody who has just joined us for the sweet edition, gluten is a water-insoluble protein. And when we beat our cake batter, what happens is the gluten protein molecules will stick together and form like cross-links. So you can see on the left-hand side of the screen, this is dough that's been kneaded a little bit. So you can see there's some cross-links between sort of different chains. However, on the right-hand side of the screen, this is after much more kneading, and you can see we've got much stronger cross-links. And what this does is it means when we inflate our our cake batter with air or gas bubbles from our raising agent, it means that we've got an elastic network around them, which means that basically they can expand, but they don't burst. So the gluten is very important in giving us a fluffy texture as well as the raising agent. Okay, so we've got our cake mixture, we've got all of the correct components, and it's risen in the oven. But towards the end of the baking process, it goes brown. And as you can see, this is the cake from the event listing. You can see the top of the cake is sort of brown, whereas the inner part of the cake is sort of a nice pale yellow. And why does it go brown? And it's due to a chemical reaction, which is called the Maillard reaction. And this is responsible for non-enzymatic browning that we see when we cook things. This is cooking anything. This is cooking if you cook bacon, if you cook roast potatoes, if you cook anything that goes brown, toast even. This is a really complicated reaction. Even as somebody who is doing a PhD in organic chemistry, I could not explain all of this reaction to you. I do not understand it myself. But we will have a good go at getting through the first couple of stages, and then we'll see kind of the result. So there's 3 stages. So to start off with, we have sugar molecules within our cake batter or our dough or whatever. There's always sugar hanging around somewhere, even if it's in the form of more complex carbohydrates. Sugars can exist in 2 forms. There's the sort of cyclic form here, and there's also this ring open form, which hopefully I can show you if I disconnect the right bond. That'll shut down there. Okay. So that is not, yeah, it is. There we are. So that is the open chain form if this bond was a double bond here. Hopefully you can see that. If this bond that I'm holding was a double bond, that is the same as if I connect it back together, the cyclic form. Okay, and that reacts with an amino group, which will be on the end of a protein, on an amino acid. They'll be everywhere, and this forms a glycosylamine. And the way that this forms is called a condensation reaction. You can see between these 2 molecules and this product molecule, we lose H2O. So we lose water, and that gives us this glycosylamine. This then isomerizes. Now, isomerization basically means we keep the same number of atoms, we just change the way in which they are arranged. So we move from the glycosylamine to an Amadori compound. And basically what we've done is we've moved 2 hydrogen atoms, one from here, and we've put them here and here. And we go via this enaminol in the middle, is how it works. But basically we move the hydrogen atoms around and we're good. Then we get to the complicated bit. So this Amadori compound can undergo so many reactions, it's not even true. And it highly depends on the conditions. So the blue arrows I've got coming off here are what happen in basic conditions. You can get fission products, you can get reductones, and in acidic conditions, you can get these sort of aromatic compounds here. And this is particularly interesting. I can sort of see how this gets to our flavour compounds. So you can get things like these furfuryls here. But trying to explain how this works, it's basically all hell breaks loose. I can't tell you how it works, but basically this makes things go brown. This gives us all of our flavour molecules. So there's lots of different molecules that will be responsible for flavour. We've got all sorts of different molecules here. These are pyrazines, pyrroles. This is an oxazole. The names don't really matter, but you can see on the slide the different types of taste that you can get from each one. And the sort of different combinations and derivatives of these parent compounds are going to work together, and that's what's going to give you distinctive flavours and enjoyable flavours. So it's going to give us this whole world of flavour from different combinations. And these are just a few things. I got very hungry making this slide about different places where this reaction would take place. It's everywhere, as I said, even in toast. So we've made our cake, that's good. What about our meringue? It's a lemon meringue cake. We need to make our meringue. So I might suggest we hand over to Jonas, who's going to make us meringue, and then I will talk about the science behind it, if that's okay, Jonas. Jonas, you're muted.
There we go. My phone is miles away. Welcome to my kitchen. I'm the head chef and owner of restaurant GL50 in Cheltenham. Today I'm going to make meringue. I'm going to do all the talking before I switch my very old and quite rubbish mixer on, because you probably won't hear anything after that. I've got icing sugar and caster sugar, and I've got egg whites in here. It's not too much to see, so I'll tell you what happens, and then hopefully we can see the nice, lovely meringue at the end of it. You've got the same amount of egg whites as you do sugar and icing sugar, so 1 to 1 to 1. I happen to have 160 grams, of egg white, and then 160 grams of sugar and icing sugar. I've also blended thyme into it because we don't do boring stuff at GL50, so this will be thyme meringue. We're going to whisk the egg, incorporating air, until it gets quite opaque, like a solid white, as far as you can take it, basically. You want to take it to the stiffest peaks you can make, and then really slowly start adding the sugar. So literally like a spoonful at a time. That I'm sure Jess will explain, but the slower the better, basically. And you have to be quite patient to make meringue. And I'm going to leave this mixing. I'm going to mute myself when it is mixing, and then we'll come back to the beautiful glossy meringue at the end of it, hopefully. The only thing I would say is if you do over whip it slightly, if you see it start to look a bit grainy, if you quickly throw in a spoonful, you will save it. Whereas other things, when you over whip egg whites, it's basically game over. And a question for you, Jess, is I don't understand what happens when you over whisk egg whites. So maybe you can delve into that when I start the machine. But it kind of, they split out and the water comes out of them, and it's not okay. So I'll stop speaking now because you probably can't hear me, and I'll slowly add this when it gets to the right point. Okay.
Fabulous. Thank you, Jonas. Should I carry on? Okay.
Over to you,
if I reshare my screen, hopefully I can get through the science of meringue by the time the meringue is made. So as Jonas said to us, can you see this, Dane?
Yeah, it's good. Yeah.
Okay, fine. As Jonas said, we have our egg whites and we add sugar slowly once we've whipped them up. So there's something special about egg whites. So what is actually in egg white? It's mainly protein and water. So as a quick reminder from earlier, proteins have a sort of specific 3D structure, and they're made up of a big long polymer which has many subunits which are all joined together, and the monomer, so the subunit or the monomer, are amino acids. And I talked about how we've got the amine group, the acid group, and then I was a bit vague about this R group. So I said there's something about there's 20 naturally occurring or common amino acids, each has a different R group. Well, I'm now going to show you the R groups. contain your excitement. So here we have the sort of 20 common amino acids with their R groups drawn out in full. I've also put the 3-letter codes underneath. These are used commonly by chemists and biologists who work in protein synthesis, because it's much quicker to write down 3 letters rather than draw out the entire amino acid. So each side chain has different properties, and we can group them together to give us different properties, and they will sort of have different chemical properties within the protein and also different physical properties. So we've got quite a large group of hydrophobic or non-polar amino acid residues. We've got a group of these sort of neutral hydrophilic or polar amino acid residues. And then we also have these ones at the bottom, which are charged, and they're charged at physiological pH. So in the pH, at the pH of our bodies or within our egg white, these would be the charges that they'll have. So we've got these sort of negatively charged ones based on carboxylic acids. And then we've got these positively charged ones, which are from sort of traditionally basic residues. And histidine here is quite interesting because this is quite easy to take this proton on and off. So it acts a bit as a shuttle. If within an enzyme, if you need a proton quite often, the histidine will act as a bit of a shuttle. So I mentioned that these properties sort of give different chemical and physical properties to the protein itself or within the protein. And it's very important in how we make meringue. So when we make meringue, sort of starting off, if you remember at my paper clips in my cup, our proteins are very globular, they have a 3D structure, and the hydrophilic side chains, that's the purple blobs, are really happy floating around in our water environment, which is represented by this big circle here. And that's because they're making lots of favourable interactions with the water molecules in the environment. On the other hand, we've got our hydrophobic or non-polar side chains. They're not making any favourable interactions with the water. In fact, it's quite unfavourable for them to be near the water. So they're actually all sort of huddled together in the middle of our protein globule, where they're shielded from the water by all the other atoms. So this is what we start with. And then when we start whisking, the physical stress of this causes denaturation. So I mentioned denaturation in the context of heating. It can also happen as a result of physical stress and also as a result of change in pH, which we'll see a little bit later. So you can see because of this denaturation, our protein strands have unraveled somewhat. They probably wouldn't unravel quite this much, but this is just a diagram. And then also when we whisk, we incorporate lots of air. And while we incorporate air, the hydrophobic side chains or R groups would really rather be anywhere apart from in our watery environment. So they sort of seek refuge in our air bubbles. So I've somewhat confusingly color coded these green. So you can see the hydrophobic side chains will cluster and point into our air bubbles, whereas our hydrophilic side chains are still happy interacting with our water molecules. The whole thing is really water, but I put some blue blobs just to show you. And what we therefore have is the protein chains and the side chains on the amino acids act to basically stabilize the foam that we make by adding the air bubbles. So we've stabilized our foam. If you were here last time, we talked about how you might stabilize a mixture of 2 different liquids, such as oil and water, when we made mayonnaise. Okay, so from there, we need to finish it off, our meringue. So we're going to add sugar. As Jonas mentioned, it's important to add it slowly. And what that is it needs to dissolve in the water in the egg white. And it basically increases the strength and the elasticity of the structure. And the sugar gets sort of trapped by the proteins. And if you add it too quickly, you can collapse the meringue. And if you add it too late, the proteins are basically all ended up tangling with each other. And it's not sort of restricted by the sugar. So then you get a sort of foamy mixture. So the sort of rate of sugar addition is quite important to meringues, but hopefully you should end up with something that's nice and glossy, which hopefully Jonas will show us as well. In terms of baking meringues, baking is normally for meringues referred to as sort of low and slow. So it's low temperature relatively, and a slow bake. They take quite a while. It's more sort of drying out the meringue and than cooking it, in that we sort of finalize the structure and we evaporate the water to give us our sort of final structure. They also involve, or should be having, a slow cool, and that means that we have no sudden sort of change in temperature, and that means that the chewy middle from our meringue, if we had a sudden change in temperature, it would shrink and it would pull on the sort of hard exterior, and that would cause cracking. So if you watched Bake Off last week, they were making pavlovas for the signature challenge, and there were a lot of discussions about when they should take it out of the oven because they wanted to avoid cracking. Jonas, are we about there? Can you give me a wave if we're there, or I can keep talking for a little bit longer? We're there, brilliant. Okay, I will stop sharing and we will, or Dane, if you could just stop me from sharing, that might be better. And we can go to Jonas.
So my very thick and slightly green tinged meringue basically goes from looking, when you whip egg whites, you can kind of see the grains a little bit, and it turns into most beautiful, shiny glossiness from all the sugar, and you want to just feel it in your mouth and on your tongue to see if you feel any grains of sugar. And if you still taste any grains, just keep it mixing. Just need it to be completely super smooth, and then when you put it in the oven overnight or whatever, here we do it overnight, you'll get perfect meringue. So it'll look the same as when they went in. If you leave it with little bits of granulated sugar, you'll just have like spots on it, or I don't know how you describe it, Jess. Kind of, it doesn't look as one, as we say. So I'm going to pipe this. We basically just dehydrate it rather than cook it. So you need to put it in very low oven at 60 degrees, or here we have a dehydrator. In whatever form or shape you fancy. The world is your oyster when it comes to meringue. You want to do a massive sheet of it that's really flat, or you want to do little Smurf hats, as we call them in the trade. Voila.
That's brilliant. Thanks, Jonas. We'll let you get piping.
Yeah.
I will reshare my screen. And hopefully this is okay for everyone. Dane, give me a shout if there's a problem. So the meringue we've talked about, there are different types of meringue. So the meringue we've talked about and how to make it is sort of basic meringue, and that's French meringue. There's also sort of two other fairly common kinds of meringue that if you watch Bake Off, you've probably heard people talk about. There's Swiss meringue. Now this is a bit heavier than French or Italian meringue. And the way that it's made is that the egg whites and sugar are sort of heated together over a bain-marie, so over some hot water, until the sugar's completely dissolved. And then it's whisked until the mixture is then cold. So as I said, this gives you a bit of a heavier texture, and you bake it as normal. Italian meringue is sort of maybe the fancy one. So this is, you have your whisked egg white as for French meringue, but then you cook it with hot, and I mean really hot. It's about 117, 118 degrees when I make this sugar syrup. So you pour your sugar syrup in while it's still whisking, and then you whisk it until it's cold. And then as you can see in this picture that I've got, a lot of people will brown it with a blowtorch or under the grill, but you don't have to, because it is actually already cooked, because you've cooked it with the hot sugar syrup. I use Italian meringue as a base when I've made macarons. So people have maybe seen macarons before. They're generally these quite expensive things in bakeries or in patisserie. They can cost about £2 per little macaron sandwich, which is quite expensive. But they're actually not that difficult to make. I went to Paris. We can possibly, people could ask me some questions about that later if you're interested. As a result of studying chemistry, I received some money from my college and went to France to basically look at the chemistry behind cooking. And one of the classes I did was on making macarons. So these are my macarons that I made. And you can see we've got this little sort of bubbly foot on each sort of macaron. It's not a biscuit, but macaron half. And then we've got this sort of smooth shell. And I'll just explain how you make that to sort of achieve that effect. So how we make macarons is you make your meringue. I make Italian meringue. I find it's a bit more stable. And then you sort of fold that into a mixture of icing sugar and almond flour. Now, when I say almond flour, I mean ground almonds as fine as you can get them, either blitzing them in a food blender, or I actually sieve mine. And that just means that you have a really smooth texture at the top of your macaron. As I said, they're a little bit faffy to make, so you don't want to go through all that effort for them to not look beautiful. And then you basically knock air out, or as the French would call it, faire du macaronnage. And what you do is you knock air out of the mixture until you can pick the mixture up on the spatula and draw an unbroken figure of 8 back into the bowl from dropping it. And then you'll pipe it. When you've got to the stage, you pipe it onto a baking sheet, as Jonas has just showed us a filled piping bag for anybody who can see Jonas's screen. And then you will drop it a couple of times on the work surface carefully. This just gets rid of any little bubbles up at the top. And then this bit now depends on where you live. You then either leave it in a warm place for about an hour until you get sort of a skin on the top of each sort of piped round. So that it's not tacky to the touch. But because I'm from the north and I live on a farm and it's freezing all the time, I can't do that. So what I do is I pop them in a sort of about 50-degree oven for about 10 minutes, and that does the job of giving me that skin. Then you bake them. And what happens, because we've got the skin, sort of the bottom puffs up to give us this little foot that you can see here and here. But the skin remains smooth on the top. You take them out, you wait for them to cool, you take them off the tray, and then you can fill them. They can be filled with all sorts of things: buttercream, this is ganache, curds, jams, you name it, whatever you want to fill it with. So macarons are a little bit sort of fancy, but they come from the same base of using meringue that we've talked about and Jonas has made for us. Also, for our lemon meringue cake, we're going to need some lemon curd to give us that tangy aspect. So a quick chat through the process of making curd. Helps if I look at the right part of my slides. So what you do is you whisk together sugar and egg yolks, and then you add lemon juice, and here they've added some lemon zest as well to help with the flavour. Then you heat that over a bain-marie very slowly until the mixture thickens, and then you add some butter to give us that. This sort of helps it to cool down a little bit and also gives you sort of a creamy texture, and that gives us our curd, which you can see cooling here. A little trick of the trade, I'm sure Jonas uses this all the time. If you've got something that might form a skin and you don't want it to, if you push down cling film right onto the top of it, it means that you don't get a skin. So this prevents it happening here. That was a little trick I learned from my time at cookery school. So what's actually going on here on a molecular level is, well, we're back to our friends, the proteins again. When we add the acid in terms of our lemon juice to the proteins, so our proteins start off as our globular chains, as we saw before for meringue. And then when we add the acid, this also is a way of denaturing proteins. So you can see that these are not quite so globular. They're starting to unravel a little bit. And what will eventually happen is they'll reach out and they'll capture other protein strands. And within that, they sort of capture the liquid. And that's cause of this coagulation kind of gives you that sort of jelly effect that you get from a curd. And then when you heat it, this further denatures the proteins. The sugar that we started off with also has an important role. It initially acts as a buffer, and it sort of prevents the protein strands from like getting to each other too quickly. Because if that happens, then you end up with scrambled eggs, which is not what we want when we're making lemon curd. So when we get to the end, we add butter. What this does is it cools the mixture, it prevents further cooking, and it also adds flavour and gives us a lovely creamy texture. Okay, so that's kind of the end of the sort of structure part of this session. But what about the taste? Like, this is probably the most important part of why we all like cake. So we're going to have a look at the chemical roots of flavour. So in our lemon meringue cake, we've got some sort of sweetness in the meringue and the cake. We've also got some sourness in the lemon curd. So we're going to start with sourness and what actually causes sour taste. So sour taste is generated by acids. I don't mean lab acid necessarily. I don't recommend drinking that. But I mean the sort of acids we have in our kitchen. So this includes things like vinegar or lemon juice. And these are acids in food. They all have the carboxylic acid functional group, which we will see a lot of in the next few minutes. So what is an acid? The chemical definition of an acid is a species which can release or donate hydrogen ions, or H+, or if anybody's very up on their chemistry, or something that can accept a pair of electrons. Don't worry about that if you don't know what I'm talking about, that's fine. In chemistry, we can sort of measure the relative concentration of H+ on a scale, and the relative concentration determines how strong it is. So we measure it on the scale called the pH scale, and this is shown at the bottom. And the way that how strong an acid is depends how far across this equilibrium we are. So if we've got lab acid, it's basically fully dissociated, fully ionized. We've got ions, so we've got pretty much 100% H+ and 100% of your anion, which I'm just going to call A-. Whereas if you've got a weaker acid, you'll have some of the material that still retains the sort of carboxylic acid functional group without releasing the H+. So you will see that weaker acids tend to lie more to the left-hand side of this equilibrium, whereas stronger acids lie far to the right-hand side. And that's kind of how acid-base chemistry really works. It's all about this reversible equilibrium. So just taking us sort of looking at the pH scale in terms of familiar liquids, hopefully less familiar to most of you would be lab acid. That's at sort of pH 0 or even negative pH if we get concentrated enough. pH 2 is lemon juice, pH 3 would be vinegar, pH 4 would be tomato juice. When we get to pH 6, that's kind of milk, and then water is neutral, and that's pH 7. So acids in food, as I mentioned, they're organic acids, and they contain the carboxylic acid functional group, which I showed you earlier. So that's this functional group. And as I said, the equilibrium sort of lies probably about here in the middle-ish. So we have quite a lot of our sort of neutral acid, and then we also have some of our ionized acid. And this is the species which gives us the, if we look at the pH indicator paper, for anybody who was here last time, this is what that will measure. The most typical carboxylic acid that you will probably have met is acetic acid or table vinegar. But there are lots of other acids in our food that are very important in giving us these sour tastes. Now, you would think that sour taste would be correlated with how strong the acid is, but that's actually not true. Sourness doesn't correlate with pH at all. And in fact, weak acids tend to be more sour. Now, this is because sort of more of the less dissociated material you've got, the more neutral species you have, it can actually cross the hydrophilic, the cell membrane. So as I mentioned earlier, if you've got phospholipids in a watery environment, you get these sort of bilayers, and these are what we find in our cell membranes. So this is the sort of hydrophobic middles, or the fatty acid tails, and this is the hydrophilic head. So if you've got things that are charged, it cannot get through this middle part of the bilayer, whereas something that is neutral has more of a chance of getting through. So that gets through into our cells, and then we perceive the taste. The character of the anion, so that would be A-, or here this carboxylate anion, also has an influence on the perception of taste, but the sort of, when I looked into this, the exact perception of sour flavour due to the anion hasn't really been studied in depth at the receptor level, which receptors are how we sort of respond to stimuli. But sort of looking sort of past that, there are a lot of common organic acids in our food. So we're going to have a quick look through what they might look like in terms of their structure. with sort of citric fruits, so that would be lemons, limes, oranges. Unsurprisingly, there's an acid called citric acid. So you can see here, this actually has 3 carboxylic acid groups in the molecule. And this acid's also present in pineapples, in strawberries, and in raspberries. So that's the acid that you can taste in lemons and limes. That's probably where you would pick it up from. Also, there's a malic acid, possibly in slightly more fruits. So we've got it in cherries, in apples, in some grapes, nectarines and apricots. Now, I've got a watermelon here. There isn't much acid in watermelon, but what there is, most of it is malic acid. And that's probably why a lot of you are thinking, but watermelons are not sour at all. There's not much acid, but what there is malic acid. One last main fruit acid would be tartaric acid. It's present a lot in grapes, a little bit in avocados. Interestingly, from an organic chemist point of view, we actually use this to determine selectivity in some of our organic reactions. So people will actually use tartaric acid in the lab. They won't extract it from grapes, but they will use this acid. There's also some common organic acids in other fruits. Succinic acid is in apples as well. Oxalic acid, the very tartness of rhubarb, that's due to oxalic acid and oxalates. Benzoic acid is present in cranberries. In blackberries, we have something called isocitric acid. The eagle-eyed amongst you will notice that the difference between this and citric acid is actually that we've just moved this OH group from here in citric acid to this carbon in isocitric acid. And iso is a bit like, is because it's an isomer. As I mentioned, isomerization, basically we have the same atoms, we've just put them in a slightly different bonding order. And also in kiwis, we have this acid called quinic acid. Looks slightly different to all the other acids, but again, it contains the carboxylic acid functional group. And this bit is kind of irrelevant in the sour taste as far as we understand it. Not only in fruit do we have acids, we get lactic acid in some dairy products such as yogurts. Propionic acid is in Swiss cheese. And we also can use acids to our advantage in vinegars, and we can use them to pickle things. And I think Jonas is going to have a quick chat with us about his vinegar making exploits. So if I can unshare, then hopefully we can all see Jonas's screen again.
Can you see me okay?
Give me 2 seconds, Jonas. There you go. That should be better.
I'm going to quickly show off my Smurf hats. I don't know if there's a more technical name, but Smurf hats is exactly what they are. So, Smurf hat meringues. So acids, certainly since when I've been a chef for 19 years, and acid is now much more prolific. Whereas before it was maybe used in things quite subtly and not so often, so why you associate maybe older food being quite rich and fat heavy. Nowadays, we season everything in the restaurant with salt, pepper, and acid. ... And as Jess just said, acid is vast. So it's not just that we just put lemon juice in everything. Here at JL50, we try and champion great British produce. So I try not to use lemon as much as possible. Obviously, in some cases, lemon juice is just amazing. But we have strived to make our own vinegar, which is probably the most exciting thing that I've done in the past 12 months, especially with lockdown and whatnot. So we have been we started with apple. Here, what have we got? This one is apple, this is pear. And then when apples and pears work really well, we just went crazy. So we've got cherry vinegar, gooseberry vinegar, which is fantastic, by the way, very much in the ballpark of lemon. I don't know which acid is in gooseberries, but the acidity is so tart because there's not actually too many acids that are like lemon juice, just for its sheer impact. Whereas gooseberries and fermented gooseberry juice, so we ferment gooseberries just with salt and blend them into a juice, and we use that a bit like lemon juice. And also raspberry vinegar, which is by far the most beautiful. And all of these stem from, first of all, making a live vinegar with apple juice, apples juiced, fresh apples from my parents' garden, and then using all trim, as you can see in these jars, You can use any trim, you can use the peelings, you could use the cores if you happen to sit and eat like 20 apples in a day. Keep all the peelings and all the core, just shove it in a jar with X amount of sugar. You can Google the exact quantities, cover it in water, and just wait. And eventually, whether it takes a month or 2 months, you'll get vinegar, which is what we did through lockdown actually. So then the end of lockdown, we had our own vinegar, which was alive. And now in all these other ones, we just add a little bit of the live vinegar, which we've cultured, and add it to all of these to add that yeast and the bacteria that you need. And then really, it's very easy. They all just turn into vinegar. So there we go. That's the end of my vinegar exploits for now. We're trying with everything. We tried vegetable vinegars as well. Beetroot vinegar is very good, and butternut squash vinegar. Who would have known?
Brilliant, Jonas. Can't wait to hear what you end up with next.
Me neither.
Okay, I will reshare. Well, nearly at the end of sort of sour compounds and acids. While we're on the subject of fruit, hopefully people have sometimes hear about some people really don't like pineapple. They say it makes their tongue sore. Some people even go as far as they're allergic to it, but it's really just, it makes their tongue sore, their lips sore. And I was a bit curious about this. It's a bit of an add-on. It's a bit of an interlude. But the reason for this is that pineapple contains an enzyme called bromelain. And it's basically a meat tenderizing enzyme, or a protease. So it basically digests protein. And it can be used in meat tenderizing. Jonas might chat to us about this later on. But basically, when people say that the pineapple juice or pineapples make their tongue sore and their lips sore, it's basically that the bromelain is already sort of acting on their to tenderize their tongue. But it's not something to worry about if you happen to like pineapple and you get that and you feel the sort of tingle and the sore lips, because once it gets into your stomach, it's not going to survive. The acid in your stomach will denature that enzyme. If you're interested in this, there's a little experiment you can try at home. If you get some powdered gelatin and then make it up into 2 little bowls as the packet's instructions, and then you maybe put some put say 10 small chunks of pineapple onto one of the bowls, pop them both in the fridge, and then bring them back out a few hours later and just see what's happened, see the difference. Maybe pop a 1p coin on each one and give them a wobble and see what happens. Pineapple's not the only fruit to contain this sort of protease or bromelain. It's also some other fruits like papaya or kiwi fruit and even figs. So that kind of brings us to the end of the interlude and also to the end of our sour tasting compounds. But what about sweet tasting compounds? Now, sweet tasting compounds are much more complicated than sour tasting compounds. The science of sweet taste is much more complex, and the sort of sweet tasting molecules that we get are much more chemically diverse, and there's a large spectrum of them. There's a variety, but we can compare them all by using something called the sweetness index. And this is a relative measure of the sweetness of the compounds compared to sucrose, which is table sugar. So here's some examples of different sweet tasting molecules. So we've already looked at carbohydrates and our sugars. Here's my glucose molecule here. Also some proteins or peptides, that's short chains of amino acids or just amino acids, can be sweet. Also, there's some heterocyclic compounds, and don't be sort of put off by the name heterocyclic. It just basically means it's a compound that forms a ring and it's got atoms other than carbon in the ring. So it's a heterocycle. So let's have a look at some of these carbohydrate compounds. So as I mentioned before, carbohydrates are polymers, and they're made up of monomers, which are the small subunits, and they can be combined in a variety of ways. Sort of, as I mentioned earlier, they can exist in 2 forms, some of these simple sugars. And you can see here these monomers, one of them has an aldehyde functional group, that's this C double bond O here, and one of them has a ketone functional group. And you may have met in sort of biology, these are called reducing sugars, and they're called reducing sugars because these functional groups basically can act as a reducing agent to reduce other things, whereas the combination of these compounds, for example, sucrose isn't reducing, it's not a reducing sugar. But as we can see here, we've got glucose and fructose. So glucose is a bit less sweet than sucrose, fructose a bit sweeter. But these actually join together to give us sucrose, which hopefully you can see. It would be more helpful if I had a bird's eye view of one of them or a side-on view of the other, but I can show you with my molecules. So here is this is my fructose, this is my glucose, and if I remove H2O, so it's another condensation reaction because we lose water, and then hopefully I can get this the right orientation for you to see. Yeah, there we go. So that should be, or maybe it's the other way around for you guys. Try that. That is the exact molecule that I've got drawn where my laser pointer is. So these sugars join together to give you sucrose, and you can see what I actually pulled off there was what should be an oxygen, but it's yellow because I ran out of atoms, and 2 hydrogens, so we've lost water. Also, another sort of sweet tasting carbohydrate derived thing. So carbohydrates are anything that is carbon, hydrogen and oxygen. It's just a name. There's also these sugar alcohols. So they're widely used in the food industry, and they are lower in sweetness. As you can see, all the sweetness indexes are lower than 1 than table sugar or sucrose, but they're also lower in calories. So you can basically use more for less calories. But it's also used in combination with other artificial sweeteners, which can be really important because it can hide the sort of sweetener aftertaste that other sweeteners can give us. And we'll see a few of those sweeteners in a little bit. So sugar alcohols do occur naturally, but normally the sugar alcohols that are put into food as a sweetener are actually made by just hydrogenating sugars. So xylitol, I've now tied up my glucose molecule, but xylitol comes from the hydrogenation of glucose. A sweetener whose name you might have heard of would be aspartame. It's got a little bit of a bad rep, aspartame, and I'll come on to that, but it's a dipeptide based sweetener. So you can see in pink here, I've highlighted, this is aspartic acid, which was one of those amino acids that we saw earlier when we were making the meringue. And then here in blue, I've got phenylalanine. And then in the darker blue, I've got a peptide bond. It's actually this is actually a CH3 group on the end, but it's derived from phenylalanine. This is commonly used as a sugar substitute. It's in all sorts of things, fizzy drinks, whatever. You will quite often look on the back of things that are sort of sweet and that will say aspartame on the back. It has had quite a lot of bad press as aspartame. There was sort of rumours or concerns that it was carcinogenic. That means it could cause cancer. But the FDA and the National Cancer Institute of America do refute this. They've run studies on this. They say that it is safe, and it is actually it has some benefits to use this as a sweetener, in that it tastes like sugar. So that's always good when we've got something that we want to use to sweeten things. It also prevents tooth decay from having too much sugar in the diet, because this won't cause quite so much problems. It also helps with weight loss, or more weight control, or maintaining a healthy diet, or even if somebody was suffering from diabetes, because this would sweeten things but not have all the calories that sugar like sucrose would have. It can be very helpful in diabetes management. And it's also very useful as a flavour enhancer or extender. So although it's had some bad press, it does seem to be that this has been investigated, and it does seem to be safe and actually has some benefits. Coming on to sort of the last set of sweet tasting compounds, which I'm going to talk about, was those heterocyclic compounds. So these are 2 examples: sodium saccharin and acesulfame potassium. This is also known as E number 950. You can see that these are massively sweet compared to sucrose, so 300 times more sweet than sucrose, and 200 times for this one. These have effectively no food energy, but the problem with using these is that they have a bit of a bitter or a metallic aftertaste. So these are the sweeteners that would be mixed with maybe the sugar alcohols or other sweeteners to try and hide that aftertaste. And in fact, one of the sweeteners that they are mixed with is sodium cyclamate. It's quite common, apparently, to have a sort of 10-to-1 mixture of sodium cyclamate to saccharin in your sweetener, sort of to hide the aftertaste, but to get the sweetness up there from the saccharin. Something interesting about sodium cyclamate is it is banned in some countries, but the EU still deems that this is safe. So I guess if you're in different countries, sometimes this will not be the additive to mask the taste, and maybe they'll use a sugar alcohol instead. So that sort of brings me to the end of my part of today, and really look forward to hear any questions from you guys. But if you have been interested in sort of the science behind cooking or the science behind baking, I'd like to point you in the direction of some of these there's some nice books. So there's Why Does Asparagus Make Your Wee Smell? Bit of a strange title, but basically addresses lots of different questions in terms of the molecules behind them. So there's things like, why does garlic make your breath smell? Is shellfish what causes shellfish poisoning? Lots of things like that. Also, Compound Interest, who are linked to this book, also run a blog. There's a lot more about sweet compounds on there. For example, there's a really nice post about chocolate, which we could talk about later if people are interested, and about different forms of chocolate and what it means to temper chocolate and how that works. And they also do sort of unrelated to the food chemistry. They do chemistry on things like pigments in terms of it getting towards Christmas. They look at what is the Christmas smell, what's in fir trees, all of that sort of stuff. Also, even they do some nice posts on sort of big chemistry events. So a couple of weeks ago, the Nobel Prize was announced, and there was a lovely post about that, sort of taking somebody from sort of very limited chemistry knowledge to be able to understand why the Nobel Prize was awarded. Also, moving on from the compound interest train, we've got this Understanding Baking book. This was one of the first books I bought when I first looked into the chemistry of cake, which was what my initial outreach events were always called, chemistry of cake, or chemistry in the kitchen. Very useful. There's a few diagrams in there. There's not very much in terms of actual molecular structures, but there are lots of information about proteins, how that works, etc. Also very useful, probably a bit harder going, the other two, is The Science of Cooking by Peter Barham, but very useful book. And with that, if anybody's looking for sort of other outreach activities to get involved with, the Department of Chemistry, my department, run lots of events, normally with schools. There's something I saw on their website to do with chirality and COVID, which sounded very interesting. Also, here I've written down about Ox Weekend. So this is Oxford Women in Chemistry. It's a sort of relatively new student-run society that I'm part of. And we're just setting up some sort of outreach events. So it would be, if you're interested, it would be a good idea to keep an eye on their Twitter about any events that might pop up. Also, they'll pop it on their website. And of course, not related to the University of Oxford as such, but please, keep an eye on everything that the Oxford Science and Ideas Festival are doing, both this year and next year, for lots of interesting things. And with that, I'd like to thank Jonas for his fabulous demonstration and chatting to us about his vinegar. Thank you very much. Take a bow. I'd like to thank the Royal Society of Chemistry because this is supported by them, this event. I'd like to thank Dane and Kathy from the Oxford Science and Ideas Festival for their sort of vision of this event and their support in me creating it. And on a personal note, I'd like to thank the University of Oxford for sort of giving me a place to do my PhD. And with that, I guess I'll hand over to Dane to sort of be the mouthpiece for any questions.
Great, thank you. So if you can stop sharing your screen now, Jess, if you like.
Yeah, brilliant.
Because I have followed the instructions, and I'm very happy to be able to share my bake that I put together last night. So let's see if I can share that. So I made my chemistry cake. They're not quite Smurf hats because I used some spoons just to dollop it onto a baking sheet, and there are some yellow and red raspberries in there to add a little bit more chemistry colour, but that's my lemon cake. So Very happy about that. I'm looking at that all evening and smelling it while you've been talking about all of these brilliant molecules.
Oh, how have you been able to resist, Dane?
Well, I may have had one of the meringues already. So we've got a couple of questions that have been put in. So someone had asked about the sweetness index, which you answered already. And they said, is there a sweetness index? So something similar to pH. I was amazed to find out there indeed was. So,
yes. Yeah, so they compare it to sucrose. I guess we have to kind of have it because there's such diversity. We have to compare all these molecules somehow. But yeah, they compare it to sucrose, which is basically table sugar.
That's the baseline. So that's one of the things about sort of chemistry and physics and basic science, is that to be able to compare across different kind of places and different situations, sometimes as a baseline or a standard. So you might be familiar with the standard kilogram or the standard measure of length. And it turns out that sweetness has its own standard too. And chemists will be exploring the boundaries of that, I'm sure. There was another question, moving on from sweetness to salt. There was a question about putting salt in meringues from someone. So there was someone thought it may have been an old culinary tale, but does vinegar and salt really stabilise a meringue? Maybe this is one for Jonas too.
It was actually me that asked the question. I thought Jess might know the answer.
Okay.
So I put in that meringue I just did, I put in a pinch of salt. It's basically out of that's what your mum says, that's what your nan says. Chefs also, you can sometimes put a splash of vinegar. It certainly doesn't stop the meringue working, but I would like to know from the mouth of a scientist if that is just made up rubbish or if somehow salt makes a meringue more stable, please.
Well, the acid thing, definitely, because that will help us to denature the proteins and unravel them, as well as the physical stress of just beating it. So I've definitely heard about putting a dash of lemon juice in or a dash of vinegar, or sometimes people say wipe the bowl with lemon juice or vinegar before you start. The salt thing, I've never actually heard that one. Amazed, and all of the times I've made meringue, never heard it.
Maybe that's flavour. I don't know.
Maybe something like that. I mean, it could also be involved. We will have there will be ions hanging around. Dane, do you know the answer?
That is exactly the answer, Jess. You're right. So
it stabilizes the ions.
It stabilizes the surface of the colloids. Wow. So one of the things that we're always trying to achieve with colloidal substances like meringue or last week we talked about all sorts of other things, but the kind of the oils and the aqueous kind of sections, they want to kind of collapse and separate out to be a big glob of water or a big glob of fat. And so if we can find a way of whisking them up and making the particles of water sit in some oil, or some particles of oil sit in some water, and just be a stable kind of colloid or an emulsion, if you like, that's great. And adding a little bit of salt puts a little bit of charge around the surface of the colloids. And so rather than having these things kind of gloop together, the charge helps to kind of repel all the different particles of liquid. So these little droplets of liquid inside a bigger space. So the salt will help to stabilize the colloidal suspension, as well as the vinegar helping to denature the protein, which, as you saw with Jess, unraveled.
Really the two things of isol, because I've only ever done one or the other, thinking they're aiding in a similar way. But really, if you do the vinegar and the salt, then you should have mind-blowing meringues every time.
Yeah, it shouldn't make a difference doing both. It should be fine.
You don't want to have as much salt vinegar as you put on your chips, though, just a tiny bit.
I'd also like to say, the books you mentioned, you didn't mention Howard McGee. This is famously the book that Heston taught himself to cook with. It's effectively a science book rather than a cooking book. And if people are interested in the science of food, this is, in my opinion, the Bible of it. Talks about the chemistry of pretty much everything. There's many additions, but it's an incredible book, just to add on to what Jess mentioned.
Is that Howard McGee? I've written that down. I've never heard of that one. I'll have a look at that. Harold.
It's incredible. It's very thin. You don't read it like
it does look a bit like a baking Bible, but we'll give it a go.
If you have a problem with ice cream, or you want to know what's going on with ice creams, this will explain in depth the main reasons that ice cream functions doesn't function. And then from that, you've got the knowledge of how to make your ice cream better, rather than like, this is how you make ice cream. It's amazing.
Good
Well,
tip.
thanks for that, Jonas.
Thanks for the tip for the book. So I was really interested in the sourness question, and there was another question in the earlier on that we had about acidity and sourness. And so the question is, why does the dissociation and going through the lipid bilayer, why is this important? Because someone was under the impression that the taste receptors for sourness are on the cell surface. So that's sort of one question, but I was really interested in how the weak acid. So in chemistry, we sometimes use the phrase weak acid and strong acid to describe the degree that the acid molecule is dissociated. But the way that the acid is dissociated determines whether or not it's a strong acid. So citric acid is called a weak acid, whereas something like sulfuric acid is described in chemistry as a strong acid. And you obviously wouldn't want to taste sulfuric acid. But Jess, just returning to the idea that a weak acid is something that can go through a cell membrane, or at least interact with a biological kind of membrane itself, could you, any thoughts on that?
So, I mean, even if it doesn't go through the bilayer necessarily, it would also be generally the receptors on our cells are not highly charged. So generally they won't pull protons so much. And also there's protons everywhere in biological systems. They're literally funneled through all over the place. So that necessarily will not just give you sour, just finding a proton. I think it's more to do with the whole acid group as a whole, rather than just the H+, which we get, which would also therefore explain why it's the sort of weaker acids where you have the whole, more of the whole functional group rather than just the H+, because especially in our cells, we use, protons are shuffled around everywhere to make our biological systems work. So if we just, everything would taste sour all the time if it was just down to protons.
And of course, even just pure water has H+ floating around. So water, as we know, is H2O, and it breaks up into OH- and H+, and we don't taste, well, we're made of water basically, aren't we? So we don't taste water so well. But if the H+ and then its companion is a complicated organic chemical, that's the bit that we taste, and it's all the long bits and the kind of the R group. So you say the residue, the R group, which is the thing that we taste. Mm-hmm. I have a question about chemistry at Oxford, if you don't mind, Jess. So someone is interested in studying chemistry, and they might even be thinking about coming to Oxford. Do you ever have any views on a good college that you might recommend, or what's it like to study chemistry at the University of Oxford? What do you get to do?
So chemistry at Oxford it's a strange beast. It's a little bit different to chemistry at other universities, and that's due to this thing called the tutorial system. So generally once a week, you will have been set for chemistry, you get a set of problems, you complete the problems, and then you go into a tutorial with your tutor. It's normally between sort of 2 of you and 6 of you, and you will talk through those problems with your tutor, who normally is a professor. So you're talking through with some of the best people in your field some of these problems, and they're helping you, and you can sort of take through it. We also do labs. There's teaching labs. So there's practical side. They're normally 2 afternoons a week. You would do practical labs. In terms of colleges, I honestly don't think the college matters that much. I was at Somerville College and absolutely loved it. Now I'm at University College because I had to move for my PhD. Due to COVID, I've actually not spent much time in University College, but I'm sure that's a lovely place too. I think maybe the way to look at colleges is, I'm assuming we have open days in person this year, is to maybe have a look round at the open days, and I think just see what you get as a feel for the place. I think generally people are pretty happy with their college wherever they end up. But if there's something you specifically want to look at, you can look in terms of which professors are there, but they might not teach. Or just have a chat with somebody who does chemistry at that college. in terms of which professors, but I could not recommend Somerville more. I thought it was brilliant for me. I never felt any pressure to be, so with some colleges they say, oh, we expect you to be, all to get first class degrees, and I sort of really credit my tutors with never ever putting that pressure on me. And then I went and got a first class degree anyway, which was lovely, but to never have that pressure was really important to me. Hopefully that answers the question, Dan.
I think it's a good question. Of course, there are loads of good chemistry departments across the country. There's Birmingham, Bristol, Manchester, there's loads of good chemistry departments. And so a lot of these chemistry departments do take an idea around this kind of tutorial system and have small group sessions, so you can get to have these conversations. And one of the things around chemistry is that you saw Jess was using these molecular models. If you're thinking about kind of studying chemistry, it's really helpful to have one of these mods. So it's such a visual subject. And so when we were talking about molecules that have a three-dimensional structure, and we talk about functional groups going inside the plane and outside the plane with these dashes and these hatched kind of shapes that Jess was showing in her diagrams, it's really important to kind of be able to understand these different kind of shapes in a way. So having this tutorial system can be really useful to get people thinking about how do the kind of molecules break down, how do they kind of fit together, and what do they look like. So there's a whole, and I'm sure there are even more new ways with virtual reality technologies and things. So I'm sure chemistry is really exciting to study as an undergraduate now. I don't know if we have any more questions. There was a little question that I was thinking about. And so sometimes when we make meringue, sometimes there's a little bit of liquid at the bottom of meringue and it sticks to the baking sheet a little bit, or sometimes there's a little kind of blob on the edge of meringue. Do we have any idea about how to prevent that?
So it's called sort of weeping or beading on the meringue when that happens. And what that is it's, how I said you have to add the sugar slowly, and how Jonas said you have to sort of either taste it or rub it between your fingers to make sure you can't feel it. And what it is if you can feel it, you have these crystals of sugar, which when you then put it in the oven, they melt. And then because they've not been captured by the protein, they basically weep out of the meringue, and you end up with these sort of quite ugly sort of caramelly coloured beads across your meringue. So that's what's going on there.
Okay, so we just need to make sure it's whisked really, really fine.
Really easy to fix at the right point, but once it's happened, there's not a lot you can do.
Right, so it's all about the preparation, and knowing the background is helpful too, isn't it? So there's another question about asking to explain a little bit more about the sweetness scale. Do we know any more about the sweetness scale?
I don't, to be honest. I don't know how they determine it, because it seems like a strange thing, but there must be a way of determining it that they have that is more scientific than somebody tasting it.
But it might well be that it is people just tasting it, like perfumes and things.
Possibly.
The human senses are so sensitive.
Yes, and I mean, there's a sweetness index, and they cross loads and loads of compounds. But I think it's useful to see in the sort of artificial sweeteners that the sweetness index is so much higher, and that's why we use these compounds as artificial sweeteners, because we need so much less compared to sugar.
Right. And then we have one more question about chemistry at Oxford. Someone's asking to know about what is the interview process like, and what do people need to do to prepare for it? Okay, so the interviews are generally, they happen in December. So I had 2 interviews, both at Somerville College. The way that mine worked is I went in, there was a piece of paper in front of me, and they started off, it was actually quite fairly basic level. They sort of started with sort of higher end GCSE based chemistry, kind of got me into the flow of it, and then built up from there. They don't ask you things you'd have never seen before. There was something I'd never seen before, and they just explained and then gave me the information and saw what I did with that. In terms of preparation, I think there's those chemistry Olympiads that schools run. Those past papers are quite good in terms of getting you to look at different problems or problems you might not have seen before. There are some outreach things that the Oxford
Oxford University as a whole run, where people will run mock interviews for people. In terms of preparing, I would say make sure you know your A-level and your GCSE work really well, know what you're studying at the moment. If you've written about a book in your personal statement, please have read it. Normally, Oxford professors haven't read that much of your personal statement. It might break people's hearts to know. But if they have and they ask you about the book, you really want to be able to talk about the book. And yeah, I think the main thing is to kind of, it sounds easy, but try and relax. I was so nervous at my interview, and I remember the tutor, he raised his eyebrows and I thought, oh my gosh, he thinks, what is this girl doing here? She's so stupid. And then, I mean, now looking back on it and looking at the question that I was answering, he was actually thinking, oh my goodness, she's got it right. Like, we've probably not had anybody do that all day. And I think in terms of preparing, the thing I always tell people who go to Oxford for an interview is, if you get an interview, no one can ever take that away from you. You are going to Oxford for a few days, and that time you get to live as an Oxford student. And yes, the interviews might be stressful and whatever, but if you come out of the interviews and you think it went brilliantly, generally it hasn't. And if you come out and you think that was awful, it's probably gone all right, because they've pushed you to the point where you basically have nothing else to give them. And they're just to assess how you think. And I think generally understand your work, do a bit of preparation looking at different problems, but honestly, try to sort of relax into it and let them guide you as much as they can, while you sort of use your chemistry know-how to sort of feel your way through maybe unfamiliar problems. I hope that helps.
Thanks, Jess. There was a question about, could we cover the science of chocolate? And fortunately, we had a whole event about this last year with Professor Elspeth Garman talking about crystal structures. Just because we're running out of time, I don't think we're going to have the chance to give that as much time as it deserves. But fortunately, there's a whole on-demand event. So I put the link in the chat box. And if you search for the surprising science of chocolate from last year's on-demand events, you can check that out. There was one little quick question about what causes overbeating of eggs and causing things to split, maybe.
Well, basically, if you overbeat your egg, the proteins will just all end up will kind of all knot together. And basically it all becomes too tight, and it just causes you don't have the foam isn't stabilized as much anymore. This is probably more your territory than mine, Dane, in terms of physical chemistry. But yeah, basically they all sort of knit together and it gets all too tight. It's the same sort of thing that happens when you add the sugar too late, and you just sort of get this sort of foamy, watery mess.
So we want a nice, smooth mixture. So I mean, the key to a lot of this is understanding that a protein. Last time we talked about gluten as being a protein. This time we're talking about egg whites as proteins, and the key is to kind of let these long, stringy like molecules tangle up just enough to suspend and trap water and oils all in the right way. But if you whisk them up too much, or if you cause them to glue together too firm, they become quite knotty and they just fall out. In chemistry, sometimes we use phrases like precipitation, like rain. So the crystals can precipitate out, or water can precipitate out if things aren't nice and smooth. And so when Jonas was blending his sugars and protein molecules together, he was doing it just at the right speed, just at the right amount of time to make sure everything was nice and smooth and uniform. Well, I think that's a good end.
It's been great. Thank you.
It's been really good. I've really enjoyed it. I'm going to sit and enjoy my lovely chemistry cake at the end of this. So it just leaves me to say thanks so much, Jonas, for being involved. Thanks, Jess, for some great chemistry confections. It's been fun to see everyone tonight, and thanks for really good questions. Thanks for coming, and we'll see you at some more If Oxford events in the future. Bye for now.